Welcome to another set of MCQs from the NEET Bio MCQ series. This is a series of MCQs chapter wise. This is helping you in quick revision of the chapter. This MCQ series is based on cell, the unit of life. Question 1. Which of the following is not enclosed by a cell membrane? Options are A. Vacuoles, B. Lysosomes, C. Ribosomes, and D. Mitochondria. Which of these do not have a cell membrane? The answer is C. Ribosomes. Explanation is ribosomes are non-membrane bound organelles found in all the cells, both eukaryotic as well as prokaryotic. Animal cells contain another non-membrane bound organelle called centrosome, which helps in cell division. So two organelles which are non-membrane bound, one is ribosome and the other one is centrosome. Question 2. The cell organelle found in plants is A. Ribosomes, B. Mitochondria, C. Golgi complex, and D. Plastids. Found only in plants. Answer is D. Plastids. The explanation is plastids are found in all plant cells and in euglenoids. Based on the type of the pigment they contain, they can be classified into chloroplast, chromoplast, and leucoplast. So had the option of chloroplast been there, this would have been easier. But here the option is of plastids. Plastids is a little bigger group to which chloroplast, chromoplast, and leucoplast belong. Question 3. Microtubules constitute A. Flagella, cilia, and peroxisomes, B. Centrioles, chromatin, and spindle fibers, C. Nucleosome, centrosome, and centrioles, and D. Cilia, centrioles, and spindle fibers. In which of these are microtubules present? To analyze this, let's understand that peroxisome is a cell-bound organelle. Chromatin and nucleosome contain DNA. So, microtubules in all the three is the option D, that is cilia, centrioles, and spindle fibers. The explanation is cilia, flagella, centrioles, and spindle fibers possess a number of microtubules running parallel to the long axis made of tubulin protein. One should revise the structure of cilia, flagella, and the centrioles before the exam. Question 4. The main function of the Golgi apparatus is A. To synthesize proteins, B. To synthesize lipids, C. Packaging of synthesized protein and lipids into vesicles, and D. Production of ATP. Golgi apparatus is closely associated with the endoplasmic reticulum. So does it synthesize or does it package? The answer is it is a packager. Answer C. Packaging of synthesized proteins and lipids into vesicles. Explanation. Golgi apparatus plays a major role in the packaging of synthesized materials into vesicles and plays important role in the formation of glycoproteins and glycolipids. Lipids are synthesized in smooth endoplasmic reticulum. Proteins are synthesized in rough endoplasmic reticulum, and ATP in the mitochondria. Question 5. The main difference between a prokaryotic cell and an eukaryotic cell is karyon means nucleus, pro means before nucleus, eu means with nucleus. If you understand the meaning of the word prokaryotic and eukaryotic, you know the answer. Eu with karyon nucleus. The options here are A. Presence of cell wall in eukaryotic cell, B. Presence of nucleus in eukaryotic cell, C. Presence of desmosomes in eukaryotic cells, and D. Presence of ribosomes in eukaryotic cells. What differentiates differentiates prokaryotic and eukaryotic cell? The answer is B, the presence of nucleus in the eukaryotic cells. Explanation. Prokaryotic cells do not have a nucleus and cell organelles except ribosomes like eukaryotic cells. Question 6. The ribosomes contain dash A. DNA plus RNA, B. DNA plus protein, C. RNA plus protein, and D. RNA plus sugar. So, what does a ribosome made up of? There are three types of ribosomes: the messenger RNA, the ribosomal RNA, and the tRNA or the transfer RNA. All of these essentially belong to the protein synthetic group. The answer is C, RNA plus protein. It's the translation of the code into the protein molecule, which is the main function of the ribosomes, the protein synthesis. Explanation. Ribosomes are the site of protein synthesis. They translate the mRNA into proteins. They are made up of RNA and protein. Question 7. DNA is not present in A. Chloroplast, B. Nucleus, C. Mitochondria, and D. Ribosomes. Where is the DNA not there? Is it there everywhere? Answer is D. Ribosomes. The ribosomes contain RNA and proteins. DNA is present in nucleus, chloroplast, and mitochondria. A careful reflect will tell us that the previous question had a bit of this answer. In every set of MCQs in the exam, there would be a set of overlapping MCQs where the question of one may contain the answer of another. It is important to be vigilant, to look out for these kind of MCQs. They help you to score and they help you to feel confident about your answer choice. Generally, the answer of one becomes the question of another. So if you are vigilant, you are able to score a little extra here. Question 8. Which scientist, based on his plant studies, concluded that cell wall is a unique feature of the plant cell? The options are A. Robert Brown, B. Theodor Schwann, C. Matthias Schildrin, and D. Rudolf Virchow. Cell wall as a feature of plant cell. The answer is B. Theodor Schwann. Explanation is, Theodor Schwann, a British zoologist, studied the plant tissues and concluded that the presence of cell wall is a unique character of the plant cells. So, he did study the cells, he did study the plant tissues and found this unique feature of cell walls. Please remember that animal cells do not contain cell wall, they contain a distinct plasma membrane. Question 9. New cells generate from dash A. Bacterial fermentation, B. Organification of abiotic materials, C. Pre-existing cells, and D. Regeneration of old cells. How does a new cell come into being? The answer is C. Pre-existing cells. Explanation. Rudolf Virchow in 1855 explained that cells divided and new cells are formed from pre-existing cells. This theory is omnis cellula a cellula. A new cell comes from a pre-existing cell. There was an era where the theory of cell generation from the organic matter was spoken of, but finally it was proved that it's only a pre-existing cell that could give rise to a new cell. Question 10. Which of the following is wrongly matched? The last question in our series. A. Cristae, infoldings in mitochondria. B. Thylakoids, the flat membranous sacs in the stroma. C. Cisternae, the disc-shaped cells in the Golgi apparatus. And D. Tonoplast, the cell membrane of the lysosomes. So which of this is wrongly matched? The answer is D. Tonoplasts. the cell membrane of the lysosome. Where does the tonoplast fit into? It's the membrane of the vacuole. Explanation. The vacuole is bound by a single membrane called the tonoplast, not the lysosome. The rest all options are correct here. In plants, the tonoplast facilitates ions and other materials transport against concentration gradient into the vacuole. The vacuole forms a major storage for the plant that helps the plant in crisis and times of need and also in times of excess. That brings us to the end of our series on cell, the unit of life. Thanks for watching. For more videos, please click the like button and share. And please subscribe to NEET Bio MCQ series. All the best. Thank you.